All right, it's 11 and the number of people are coming in. Daniel will be admitting people from the waiting room. We have this second layer of security so that nobody unregistered will come in and disrupt the whole meeting. Good morning, afternoon, and evening, everyone, wherever you are logging in from. My name is Arthur Luher. I'm the Associate Director at the DGRC here. We collect and distribute Drosophila DNA reagents, plasmids, and cell lines. We help people use the reagents and also develop new ones. Director Andy Zelhoff, Associate Director Daniel Mariappa, and myself, database specialist Chris Hemmerich, and staff scientist Johnny Roberts make up the current core DGRC personnel. And we would also like to take this opportunity to congratulate Dr. Chris Klug, one of the DGRC's founding members, on her recent retirement. So today we are gathering here to remember the contributions of Dr. Lucy Cherbas, whose legacy continues to shape the landscape of Drosophila cell culture research and beyond. Lucy was a senior research scientist here at the Department of Biology at IU since 1985 until her retirement in 2016. In 2003, Lucy played a pivotal role in the founding of the DGRC here at IU, where she significantly expanded the availability and the utility of the Drosophila tissue culture cells. Her expertise and dedication left a lasting impact on the scientific community. She passed away in October 2022. And to honor her legacy, DGRC started this seminar series, the Lucy Cherbas Seminar Series, to have experts from the fly community share in a seminar format how they use Drosophila cell cultures to address basic biological questions. During this seminar, please raise your hand if you have questions or use the chat box anytime during the talk to pose your questions. Our speaker will be happy to take questions during the time, and we will help to moderate the Q&A session. So it is now my distinct pleasure to introduce our seminar speaker today. Our speaker today is the director at the Drosophila Research and Screening Center, Biomedical Technology Research Resource, located within the lab of Professor Nobel Perimon at the Department of Genetics, Harvard Medical School. In her capacity, she leads and oversees novel research initiatives and high throughput screens using insect cells. She received her BA with honors in biology from Wesleyan University and went on to earn her PhD from the University of Colorado, Boulder, where she did her research on Drosophila oogenesis under Prof. Robert Boswell. She then served as a postdoc fellow under the guidance of the late Professor William Galbert, and then transitioned into a role as a scientific liaison at the Harvard Institute of Proteomics. She has authored over 70 research reviews, protocol publications, and addressing fundamental questions in biology, high throughput screening, molecular genetics technologies. Her editorial endeavors include co-editing Drosophila Cells in Culture, the book that you are seeing in the slide, and authoring the widely acclaimed book First in Fly, Drosophila Research and Biological Diversity Discovery. This book was recognized as one of the best biology books of 2018 by Forbes and the Smithsonian Magazine. So, and beyond her scholarly pursuits, she's also committed to scientific outreach and community engagement. And here to discuss high throughput screens in insect cells, please welcome Dr. Stephanie Mohr.
Thank you, Arthur. Let me switch out the slides. Looks good. So I want to thank you for that introduction, and I want to thank Arthur and the rest of the organizers at the DGRC for the invitation to speak. And I too want to start by acknowledging that we're meeting here today to honor Lucy Cherbas. I interacted with her several times before her retirement in 2016. As a scientist, I can say that my group has benefited greatly from the data generated under Lucy's direction. And then more broadly, I would say that we have all benefited from the precedent she and others set of combining a scientific career with a focus on technology and community outreach. Hers is, in my view, a lasting legacy, and I'm very pleased to take part in today's event. So as Arthur said, I'll be talking today about high throughput screens in insect cell lines in terms of technology, assays, and relevance. So the first thing I wanted to do, however, was to spend a couple of slides just explaining the name and our current mission. I think most of you will probably be more familiar with our original name of Drosophila RNAi Screening Center. We now, in 2019, became the Drosophila Research and Screening Center to preserve the acronym, but expand the technology. And we formed as an NIH-recognized biomedical technology research resource. So BTRRs under NIH direction have a number of technology areas in which they do development and improvement of those technologies. And the areas in which my group focuses are cell-based technologies and screening, next generation in vivo technologies such as the TRIP collection of CRISPR flies, and innovation in new Cas forms in vivo, and in the area of Drosophila proteomics, such as development of proximity labeling technologies and nanobody-based nanotagging. So I'll be exclusively focusing on the cell-based tech and screens, but I want to point out that all of these efforts also integrate bioinformatics. So we are blessed to have a big bioinformatics team, well, not big, but very effective bioinformatics team that supports all of these efforts, from reagent design and experimental design all the way through to data analysis. So the model by which the DRSC-BTRR operates is one that I would describe as having an iterative cycle of improvement as our goal. So technologies ideas feed into the DRSC-BTRR from the Paramount Lab, from technology experts in other fields and working in other systems, and from other sources. When we adopt a technology for development and improvement, we then partner it with specific biomedical or biological projects that were proposed by community members. And that pairing of technology with external collaborators' projects ensures that we are developing technologies that meet real needs in the community. And it also ensures that we then get feedback data back from those groups that allows us to further improve the technologies in order to meet those needs. When technologies are considered mature, then we then disseminate the information through outreach training. We also deposit materials to repositories, including the DGRC that's hosting today. So since our formation as the DRSC-BTRR in 2019, we've engaged in a couple dozen projects with labs based in 15 US states and in other countries, and also with PIs at every career stage, which is exciting and fun for us. So today I'm going to focus first on giving an overview of the RNAi cell screening technology that's our long legacy, then introduce the current state-of-the-art of cell screening here using CRISPR technology, and finally end by talking about how we're extending this CRISPR cell screening platform to mosquito-cultured cells and tick cells. So I want to start by making what may be a both obvious and somewhat surprising statement that culture cell studies are not the end goal. So one of the things that we try hard to do is to always have in mind that the studies we're doing are in a context, and we're basing these studies on known knowledge, but the goal is then to translate these studies to in vivo and then perhaps to applications in the long term. And sometimes this is a path that's going to take decades, but I think you'll see in some of the examples that I'll provide that this can also be something that plays out within one project. And one of the things I'm hoping will be the case in the audience here today is that some of you will begin to get some ideas about how a cell-based study might fit into what I would guess for most of you is a largely in vivo focused effort within Drosophila. One of the reasons, or some of the reasons, that we turn to cell studies is they offer a relatively uniform cell type and therefore easily scalable. So you can grow a lot of a cell that's going to have a uniform response to a treatment or to some kind of condition, and that provides an opportunity for discovery that could be much more challenging in an in vivo format if initiated there. So the Drosophila RNAi Screening Center was founded by Norbert Perimon in 2004, building on the success of screening in his lab. For example, they started with about 1,000 gene RNAi collection and looked at cell morphology. They then went to genome-wide scale and looked at growth and viability genes, and then turned to other types of assays, including host pathogen interaction assays. And between 2004 and present day, when we're, I will say, winding down this technology, there have been more than 200 large-scale RNAi screen projects with impact in many fields. I joined the group in 2008, and that's about the time the transgenic RNAi project first started. We kind of had a functional merging of our website and our activities in 2016. As I said, in 2019, we began as the DRSC-BTRR. And as an unfortunate accident of fortunate funding, the NIH is probably going to require us to change our name one more time to the DRSC-BTDT. But we'll try to always preserve the DRC part to keep things as simple as can be. So one of the motivations for that change in 2019 was the fact that Raghavir Ramviswanatha showed, who was a postdoc in the Perimon lab, showed in 2018 that it's possible to do genome-wide CRISPR cell screening in Drosophila cells. And I'll talk about the technology that he developed. In 2021, we reported that we're also able to do this in mosquito cells, and I'll talk briefly about that as well. And then there have been, on the heels of that, a number of both published and now many unpublished screens that are looking at various applications of this technology, in particular using screens for resistance to a treatment or a selection. And I want to also highlight that in 2013, Baolong Chia, also a Perimon lab postdoc, demonstrated that CRISPR activation screens are possible at genome-wide scale in fly cells. So there are currently about 50 cell screens that have been completed or are in progress. And from my outreach to the community, I have the impression that this is going to further blossom in the future. So I want to start, one of the things that's going to be obvious in the difference between the RNAi screen platform and the CRISPR screen platform is the reagent. But as these screens are done here at the DRSC, there's another key difference that affects the biology of the assay, what you can discover and how you might discover that. And that is that our RNAi platform is done in an arrayed format where you have an individual reagent dispensed into an individual well, that we then dispense cells into those wells, do some kind of well-by-well assay, which might be imaging or could be something on like luminescence on a plate reader. And then to associate phenotype with genotype, we simply look up okay, this, well B3 was a hit in my screen as positive for the phenotype. And you look up in a database or a spreadsheet what reagent and thereby, what gene was meant to be targeted in that well, and thereby associate phenotype with genotype. By contrast, the way that we are doing CRISPR screens is in a pooled cell or a pooled cell screen format. So in this case, the reagent library is introduced en masse and in bulk. So cell by cell, they might have different knockouts, and we don't know, which cell next to the other has which. We then do some kind of assay that separates two populations of cells, basically wild type cells, from some phenotype of interest. And then we have to amplify the guide RNAs, and that serves as a barcode to let us then associate the genotype with phenotype. And so either depletion or enrichment of that barcode becomes the readout that lets us identify the association between genes and phenotypes. So focusing on the arrayed screen format, it provides excellent support for assays that include total luminescence or fluorescence. All kinds of subcellular level image-based assays are possible with this format. We've supported some screens that, in fact, went as far as doing qPCR, and even a screen that did high-throughput RNA-seq. A disadvantage of this format is that it requires specialized equipment. So it's fun for us because we get to play with robots, liquid handling robots in the lab. But overall, for the community, it has a disadvantage in that both to produce the RNAi reagents, as we do here at the DRC, but also to perform the screens, which ideally would be done in a home lab, you do still need this specialized equipment in order to dispense cells, dispense reagents, read out the screens, and so on. It does have an advantage, though, of supporting, as I said, easily supporting high complexity assays. So with this high complexity assay format in mind, we've long had a goal to generate cell lines in which different organelles are painted with a fluorophore. We imagine that supporting image-based screens. We also imagine that being more broadly useful in the community. And there have been two very different efforts to accomplish this goal using the technology available at those two different times. So the first time that we had tackled this at large scale was using the mimic transposon approach developed in Hugo Bellin's lab. So in this case, we started with a plasmid that has ATP sites flanking M-cherry, and M-cherry is in this format has splice donor and acceptor sites on either side. So it's acting as an artificial exon. It has no promoter of its own. So from free-floating plasmid in cells, you would not expect to get M-cherry, but if it integrates into the intron between two coding exons, then it could become part of a translated fusion protein. And in some cases, if the localization signal for a given protein is intact in that setting, it might be localized to a specific region. So Stella Lee in the lab at the time took this on at high throughput, did a lot of single cell cloning, and was able to identify a number of cell lines, a few which I'm highlighting here. And we did, in fact, use this trap approach to identify fluorophores that light up the cleavage furrow, Golgi ER, and other locations. Now, I mentioned that this is flanked by ATP. For the goal that we had in mind, this was inconsequential and really just an accident of the fact that we didn't want to re-engineer a plasmid that was originally developed for in vivo use, where the Bellin lab was interested in vivo to be using those ATP sites for recombination mediated cassette exchange. This happens to be to turn out to be a happy accident for us. So yeah, I'm going to highlight this NPT5 cell, and you'll see that it comes up in the talk later. So of course, when CRISPR comes along, we can take on this same task of sort of painting organelles and cells at a much higher throughput, or I'm sorry, at a much more precise way and a much more efficient way, because we now were able to do CRISPR knock-in targets that we knew had specific localization patterns. So we took on a number of them, and most of them were then localized to the expected component. And some of them we tagged with GFP, and others we Justin Bosch tagged with using the CRISP paint method with M-neon green. So I'm listing these here, and these are available. You can see lists of them at our website, and you can find them at the DGRC as well. And I can't resist the opportunity then to show you a video of one of these. So this video was taken by my colleague, Jonathan Zirin, and reported in Conca et al. In 2019. This is GFP fusion to Polo created by CRISPR knock-in. What you can see is that Polo has lighting up kinetochores, and thus has a very cell cycle regulated discrete subcellular organization. So I tasked myself with kind of summing up what we got out of more than 200 RNAi screens. And I have to say that emotionally and scientifically, this is not an easy task to do. So what I'm going to say is that I came up with a couple of examples of general categories that represent general categories of screens that had impact. So I'll start with some of the more recent cell screens using the RNAi technology. So it was recognized that we could use CRISPR to make knockout cell lines and then do a genome-wide RNAi screen. And so a couple of those were done by a couple groups. And the overall goal there was to identify perturbations that resulted in synthetic lethality. So lethality that was associated with knockout of, say, a tumor suppressor gene, but not associated with the wild type cell background. Those then become potential, those when they're conserved became potential drug targets for translational applications. Another very broad category in which there was a lot of RNAi screening that led to new findings was in general in signal transduction, and in some specific examples in immune suppression. So I wanted to highlight a couple of these studies that had impact, including the first identified in vivo mediator of what's known as the hypercapnic immune suppression response from the Vitale lab at Northwestern, and other studies of immunity. There was also a kind of tour de force screen, in my view, that was done by another Paramount Lab postdoc, in which they they detected the nucleolus within the nucleus and did a screen to look for mutant phenotypes in using the RNAi platform, and then did a comparable screen of a knockout collection in yeast, looking at the yeast nucleolus and identifying genes in yeast that led to a disruption of the normal distribution of the nucleolus, and thus were able to combine those results and come up with a list of highly conserved nucleolar regulating genes. I mentioned that Sarah Cherry in the Paramount Lab early on used that platform to do host pathogen screens. That was extended by many community members, including screens of Francisella tularensis and dengue virus. And then another, a final broad category that I'll mention was identification of ion transport channels. And the two examples I'm showing here was identification in the lower redundant cell, Drosophila cell system of what turned out to be conserved ion transport proteins. And two of those also turned out to be human disease genes. So I'll transition now to CRISPR, but I see that there's one comment in the chat. Do we have a question already, or should I keep going? I think that was a thumbs up to keep going, so I'll go with that. All right. So I talked about the pros and cons of the format in which we do RNAi. We can also mention that the reagent leads to knockdown and that it has some known off-target issues. I'll also mention that the typical screen scenario was to have a 4 to 5 day incubation of the reagent with the cells. So that combination of 4 to 5 day incubation with knockdown made it possible to capture, say, subcellular phenotypes observable in cells, even when the gene would eventually lead to death of the cell. So this was another feature of that format and timeline. By contrast with the CRISPR, as I said, we're doing this in a pooled format, introducing library at random in bulk. The big plus is that this is supportable by standard lab equipment, and does mean that we have to adjust our thinking to assays amenable to pool format, and I'll talk about what some of those assays are and how we can push the limits there. The timeline is also very different. So this is going to affect the biology of what's being actually queried. And so the timeline of these screens is typically 4 to 6 weeks of outgrowth. And again, because it's a CRISPR, we're looking at knockout, the way that we're using the CRISPR system. And that has the wonderful benefit of better precision than RNAi. So I want to go into some detail. Again, I'm hoping that some of you are going to get excited to do this. So I want to make sure you're really understanding how we're doing these screens. So it starts with design of a library and building of that library. So we do the Claire Yunwei Hu and her team designed the first generation library with 6 to 8 guides per gene coverage of the approximately 14,000 genes in the Drosophila genome for a total of more than 85,000 guides in that library. Now that can be then synthesized on a chip and cloned into a backbone vector. Again, we're doing this in pooled and bulk, so it's not an easy. We need to maintain that complexity, but it's a very different task than trying to do that in arrayed format. Those plasmids in which the U6-Pol3 promoter is driving expression of the gene then get integrated into the cells. This is where our NPT5 cells became useful to Ram as he developed this platform. So again, Ram Viswanatha recognized that he needed a way to integrate the guide plasmids into the genome so that they are retained, and every time the cell replicates, it's copying that information, and that makes it become a barcode that we can read out later in the assay. The screen assays themselves are varied. Here are 2 basic examples of the types of screens we were doing earliest on in this pooled format. So in one, you can imagine that this cell represented in blue has a guide targeting a gene that's required for growth or viability that over time is simply going to be lost for the population. So you can look at genes that are depleted or dropped out, as we say. So we can do dropout assays to uncover genes required for viability and growth, either in a given even in wild type cells or in some other context. It's also feasible then to take, the cells that remain alive and treat them with some toxin and ask if we can identify, say, cells that are resistant that would be able to continue to proliferate despite the fact that a wild type cell without that resistance would normally perish. Then, of course, there's this data analysis and application or data analysis step, and I'll describe that in a later slide in a little bit more detail. So I really want to bring home this idea that key to this technology, and this is again something that Ram worked out, is that we need to integrate the plasmid into the genome. So if that didn't happen, then we would have no way to, in the long term, associate the guide and thus the gene that was being targeted with the phenotype. But integration makes that possible. In mammalian systems, this is achieved using lentivirus. That system simply doesn't work in these cells. And so what Ram recognized is that the Phi-C31 integrase in the presence of these ATP sites in the cell line and ATB in the plasmids being delivered as the library make that integration possible. So with regards to data analysis, then that makes it possible for us to take a time zero or another control set, an experimental set, pellet the cells, extract genomic DNA. In the two populations of genomic DNA, we're going to have different distributions of those guides, again, because some of the cells have died from having the guide that targeted an essential gene, for example. We can amplify those then with PCR and read this out using next generation sequencing. And the output then becomes a rank order either of guides or of genes and allows us to compare control and experimental sets. So the first screen that Ram did was to look at one of these dropout screens to identify what guides, as represented here in yellow, are depleted from the endpoint as compared to the beginning. He then moved on to doing a number of resistance assays, and I'm going to highlight one of them. So the particular biological question that this queries is one of understanding the toxins that are made by bacteria that are symbionts of entomopathogenic nematodes. So these are nematodes that attack insect larvae. They enter the larvae. They then release the bacteria that have toxins. Essentially, this turns the inside of the larvae to a wonderful nutrient soup for the growth and support of those nematodes. They then reproduce and lyse from the larval corpse. So there was a known toxin, one of these TC family toxins, for example, from one of these bacteria symbionts of the entomopathic nematode called Photorhabdus luminescens. And the Min Dong's lab at Boston Children's Hospital had a number of reasons to be interested in TC toxin. They tried mammalian screens, but there was not a known receptor. And so they turned to the insect cell platform, and Ying Zhu in Min Dong's lab collaborated with Ram to do this study. So one of the early tests they did was to see what happens to the screen-ready S2R+ cells that have this mCherry and have Cas9 and so on when you treat them with the toxin. And what happens is with increasing concentrations from left to right here of either the P. luminescens TC toxin or TC toxin from another species of bacteria, we can see that the cells swell up. This gave them an opportunity to do an assay in which they took advantage of the fact that that the wild type response is going to be this cell swelling, and that any resistance in knockouts that confer resistance, so for example, knockout of a receptor of that toxic protein, could result in maintaining that small size. So they literally put these cells through a strainer of 30 microns. The larger, normally responding cells are going to burst, and only these smaller, potentially resistant cells are going to get through that strainer alive and be able to proliferate. We can then compare that end population with the beginning again to identify the data. So what we're seeing here is data from Ying and Ram that we're on the x-axis, we're seeing one replicate, and the y, another replicate from the same screen with PTC toxin. And they identified the strongest hit here in both screens, replicates, is Visgin, a fly gene that's a cell surface protein, and they were able to show that this is likely to be the receptor for the toxin. It's the toxin. The model is that the toxin directly binds to the Visgin protein on the cell surface, and then this becomes internalized. One of the things that was really nice is that in addition to the receptor, they also were able to identify what are very likely the components required for internalization. And in this case, with the PTC toxin, they identified, for example, members of the early endosome formation pathway. We were interested to know whether these are going to be specific effects or general effects. And the example I'm showing here, I think, is going to answer that question, at least with regards to the bacterial toxin screens. So another thing that Ying and Ram did was to compare results with PTC here on the y-axis now, where we see Visgin as a top hit, with results of a comparable screen using another toxin. And what I hope you can appreciate here is that a different set of genes are hits in the MTC screen. And so we're able to identify a very different internalization pathway and thus, confirm that we're getting very specific results and learn something interesting about the biology of these toxins, the differing biology of these toxins. And I would further argue that, if your goal was to identify all the things required for early endosome formation, this is also a reasonable way to get new candidates for that pathway. So they were interested to see the impact of this Visgin in vivo. So they generated Visgin knockout flies. They then touched a needle to P. luminescens bacteria and used that to poke into flies, and asked about fly survival and the number of bacterial colony forming units that are possible to recover from the flies that had been infected in that way. So if we focus here on the solid lines in black, we see what happens when a needle that has been exposed to P. luminescens pricks a wild type fly. You see that the flies begin to die within just a little over a day. By contrast, in red, we're seeing here the results with the Visgin knockout fly. So even here, we're not using just the toxin for which they used in screen, but the full bacteria that likely has other factors as well. Even though that's the case, they nevertheless see a decrease in sensitivity to the toxin, and that's evident here as well with a decrease in the number of colony forming units. So I want to sum up some of what's been learned so far from these CRISPR screens. So as I said, Ram started with genome-wide survey of essential genes, and you'll see later in the talk how that we made use of that data set in a new way. They also include identification by Ram in collaboration with the Yamanaka lab and in vivo validation of what turned out to be a transporter of ecdysone hormone, which had previously been thought to simply diffuse into cells. I gave you the example of showing how they're identifying bacterial toxins along with the Dong lab, and in collaboration with other labs, Ram has identified receptors and internalization mechanisms for additional bacterial and fungal toxins. We've also conducted a screen using tunicamycin, which affects glycosylation, to identify potential drug targets for a rare congenital disorder of glycosylation that is in some ways mimicked by that tunicamycin treatment. And I mentioned that we've had this technological advance by Balong Chia, who showed that CRISPR activation screens are possible. So I want to transition here to thinking about what we can learn from these screens, again, with this idea of trying to put some ideas in your head or encourage your own creativity and how we might be able to continue to use this platform efficiently and effectively. So these dropout screens that identify essential genes, of course, are going to show us what are the fundamental cell machinery involved in these cells. Application of this type of screen to cells such as those that Amanda Simcox described in her talk, the first in this series in the fall, which is available online and I highly recommend, might be one route to identify cell type specific requirements within cells. And I think even though as Drosophila biologist, I feel like there's been a large part of my career in which I was in the business of keeping flies alive, there are a lot of reasons that humanity is sometimes interested to kill flies or their cousins in the Diptera order. And so one of the things that I think is exciting about these dropout screens is also the potential for identification of targets for species-specific insecticides. And I think it speaks for itself that that might have ecological benefits compared to broad-spectrum insecticides in use now. So with regards to resistance to toxin treatments, I mentioned that they can be used to identify receptors, transporters, and targets that are directly related to the treatment, but also to uncover components of cellular pathways and activities that are related to those treatments. I showed you example of endocytosis through the toxin screen, mentioned that we've also looked at glycosylation, and we looked at some specific signaling related pathways in this way. Another area in which we think that there's going to be impact is looking at host genes required for pathogen entry, internalization, and replication. And then we also can return to this idea of synthetic lethality that I told you was done using the RNAi platform, but now using the CRISPR pooled knockout platform, in which we can compare the outcomes with wild type cells to outcomes with knockout cells, again, for example, with a knockout of a tumor suppressor, in order to identify perhaps targets for cancer, and more broadly to uncover genetic networks. So some of these screens are also underway. And then an area of active technology development in our group right now is to develop reporters that are fluorescent, either on or off in response to some activity, for example, that could then be used to use that fluorescence as the output. So using fluorescence activated cell sorting then as the way to separate the phenotype negative and phenotype positive cells. So we're looking at things like metabolic sensors and pathway readouts in a fluorescent mode. And we hope that this should uncover metabolic regulation, pathway components, and further expand the toolbox of assay types and the complexity of assay that can be developed. So what's next in terms of Drosophila cell screenings includes to expand those assay technologies. We'd like to, we're in the process of expanding technologies in terms of the Cas system itself. So there are Cas proteins that target RNA. There are fusions that act as base editors, and we're exploring their use in Drosophila cells, partly for to create new screening platforms that might have other advantages compared to the knockout platform, but also with the idea that we can use this as a quick way to test prior to going in vivo for our in vivo technology efforts. And then we're additionally interested to establish this screening platform in additional fly cell lines. So to date, the Drosophila cell screens have been limited to S2 cells, but that's not a limit that we have to adhere to. So I'm going to transition now to talking about how we have used what we learned from Drosophila to dive into non-model species. And I'll start with the species group that we started with, which is mosquitoes. So like Drosophila, these are dipterans. They're close cousins of the fly, one might say. They are similar in genome size, similar in number of protein coding genes. If we compare the genes in Anopheles with the genes in Drosophila, we can find a Drosophila ortholog for, depending on the stringency by which we ask that question, between 70 and 80% of the genes. So again, an indicator that they're close relatives. If you do a search in PubMed with Drosophila as your search term, there are more than 100,000 results retrieved. But speaking to the relative understudy of non-model species, and Anopheles specifically, there are only about 19,000 PubMed results. So I use this as a proxy to say there's a lot to learn about mosquitoes. And of course, the human health relevance, I hope is obvious to the audience. The Anopheles gambiae is the vector for malaria. We've also been working with cell lines from Aedes and Culex that transmit other diseases. So based on the technology that Ram established in Drosophila, we can essentially develop a checklist of what we need in order to establish screening in some new cell line. That includes genome-wide our guide design. We need a cell line that has ADP for the recombination-mediated cassette exchange to introduce the sgRNAs. We need a source of Cas9 in order to have efficient cutting, and we need Pol3 promoters because we need to be able to express the guides from that promoter. So in order to develop genome-wide guide designs for mosquito cells, we turned to the data that Ram had got from the initial dropout screens. So you can imagine that if I give you this imaginary gene data or screen data for gene A, let's imagine that one of the guides for gene A was like the top hit, and the next one was, the second to top hit. So you can see that overall in our in the algorithm that the team applies, gene A would have scored at the top or close to the top as positive in this assay and scored as an essential gene. But that doesn't mean that all of the guides work or have that phenotype. So it's likely that in some cases, here I'm giving this made up example of a couple of guides that would be scoring in a range that we would kind of consider a non-hit. So we can, broadly speaking, say that we have good designs and we have bad designs. And we now had, thanks to Ram, with this data set where we could take a look at a large number of what could be categorized as good and bad designs. So Pierre Mercart joined our lab as an intern with experience in machine learning, and together with Claire-Yan Hu, they used the version 1 library data. They identified these good and bad from the gene level hits, and then they applied a machine learning approach to identify position matrix information and assign an overall score to guides. So the position matrix, what I mean there is, like we could say at position 4 of the 20 base pair guide, you have an advantage if you have this particular base and not these other particular bases. So we're then able to apply those rules and those scores, not just to Drosophila for a version 2 library that has proved its merit in Ram's hands, but also apply that to the design that we then chose for mosquitoes. So Claire used that machine learning approach to assign scores and prioritize what based on that data at least were likely to be good. She also was able to benefit from comparison of available whole genome sequencing from cell lines that we were using for mosquitoes and from a wealth of wild population database information in order to exclude things that were likely to have SNPs. She developed genome-wide designs then for Anopheles, Aedes, and Culex, and four species of mosquitoes altogether with two different species of Aedes genus, and makes all of this information available at a resource called CRISPR Guide Express. So we can check one of the box. What about the other two? So I'm going to just, in the interest of time, tell you that we were able to accomplish the ADP site in the same way. So we used the Minos gene trap approach to add ADP, mCherry, ADP into these cells, into the 4 different species of mosquito cell. And this is work by Enzo Mameli together with Ram. And we also, Ram and Enzo also made those cell lines then positive for Cas9 through transient transfection and identification of stable cell lines with Cas9. So that left the problem of needing a Pol3 promoter for the plasmids with which we actually build the library. So the approach that Enzo Mameli took was to look in the reference genomes for these mosquitoes, look for small nuclear RNA genes that were annotated, look upstream of those for consensus for the U6 sequence. And using that approach, he was able to find 2 or 3 candidates for different species. He compared those among each other and with Drosophila in various cell lines, in all of the cell lines we're working with, and perhaps not surprisingly found that like the within species guides or U6 promoters were typically the most effective for a given cell species. So he was able to identify and validate those U6 promoters, and it made it possible then to build a library that was going to work. They started with a focus library rather than a full genome library. And in this case, we're looking at data from Anopheles cells. So this was by our measure, a moderate screen. So there are only a few thousand things being queried here. And we can just take an example, as an example, a treatment of the Anopheles cells with ecdysone normally arrests their cell growth. But if you knock out the receptor, that's the ecdysone receptor ortholog, like not surprisingly, then scored as a resistance factor. But you can see in the data for very different, for compounds that have no relationship to ecdysone signaling, they score as non-hits as one would expect. And then the opposite was true for these other treatments. So validating that this is working. So we've currently now also completed production of genome-wide screen library and a couple of initial genome-wide screens in Anopheles. And Enzo Mameli and others are doing this in collaboration with Ryan Smith at Iowa State. And then looking to the future, we want to establish genome-wide screening libraries and platforms for additional mosquito species, expand the available assays in ways parallel to what we're doing in Drosophila, and focus we expect to focus on host pathogen interactions in these studies for the perhaps obvious reason that we're looking at mosquito cells because they are insect vectors of disease. So finally, we were encouraged by that and thought, well, what else should and can we try? And so we have more recently been extending the platform to the deer tick or black-legged tick, Azotes scapularis. So I'm sitting here in the New England region where Lyme disease is well known to be a problem in this area, and it is carried by this particular species of tick in this region, Azotes scapularis. And Azotes scapularis in particular and ticks in general are of increasing human disease concern. So this species carries not only Lyme, but is also capable of transmitting about a dozen other diseases that can affect humans. They are relatively understudied. As you can see, we're dropping now to only 9,000 PubMed results with the genus name Azotes. They're also not the greatest if you're thinking about in vivo genetics. They have a much larger genome size, so about 10 times that of Drosophila. They have a larger number of protein coding genes, a larger number of chromosomes, and they're more different from Drosophila than mosquitoes. And I think possibly argue that they're more different than humans are from Drosophila. So if you take the genes in Azotes scapularis and ask for what percentage there's an ortholog in Drosophila, that's a best somewhere between 30 and 44%, depending on how stringent you are with that. So there's a lot of intellectual space and a clear need to get information about this human health relevant species. So we were taking on a higher risk, but we had a couple of things that were in our favor. So, and just to bring home the idea of risk here, we take a lot of things granted. It's one of the things we have learned from these studies. So for Drosophila melanogaster, we all know this life cycle is about 2 weeks. By contrast, for this tick, it's about 2 years, and you can push that a little faster in lab settings, but still is like a scary time cycle to think about, say, a large-scale in vivo genome-wide screen. In addition, the lab food that we feed our flies is cornmeal with a bit of yeast and sugar. By contrast, Azotes scapularis absolutely requires a live vertebrate blood meal at each life cycle stage. So if you're keeping these ticks, you're also keeping mice and or rabbits. The genome of the fly in a context like this, we can describe as extremely well annotated, whereas Azotes scapularis is benefiting from application of the newest next generation technologies for sequencing RNA and DNA. And there's been some recent advantages, but it's still fair to say that particularly compared to Drosophila, it's not as well annotated. And another thing we take completely for granted for Drosophila is that we have Pol2 promoters to express protein coding genes, and that we had Pol3 promoters as well. Whereas for Azotes scapularis, there are actually few characterized Pol2, either exogenous or endogenous promoters. And at the time we initiated our studies, no Pol3 promoters. But as I said, there are a few things that made us optimistic and willing to take this on. One of them is that there are, in fact, cell lines for ticks, right? So we didn't have to start from complete scratch. So this is the work of Ulrike Munderlo and Tim Curti. They isolated back in the 1990s a number of different cell lines from Azotes scapularis embryos. These require a custom media formulation. They cannot be single cell cloned, but luckily for us, they are amenable to FACS. They're considerably slower growing than Drosophila cells, so we had to kind of adjust all of our expectations in terms of timeline. And if you weren't already creeped out by my talking about ticks, I will mention that these cells, we have a dedicated incubator at 34 degrees, which is mimicking skin surface temperature of like our skin or another vertebrate. Another piece of information that made us feel like this was a risk worth taking is that work from Julia News's lab had shown that the CRISPR-Cas system is functional in Azotes scapularis ticks. So the Julia News lab did some beautiful studies in which we were able to show that they can remove an organ that normally coats the egg with wax. That makes it then amenable to injection, which the wax-coated egg is not. But they nevertheless can develop, so they're able to inject the CRISPR-Cas machinery into those eggs, and we're able to observe gene editing events. So that made us optimistic that, okay, so this is likely to work at some level. So we can go through our checklist again. And I'll simply tell you that Claire and her team was able to apply the same kinds of rules and develop a genome-wide guide design resource. Matt Butnaro and a team here have taken on the next steps, including generating a cell line with ATP. So we gave a try with the Minos approach, but at least in the attempts that we didn't find that it was working. So Matt and his team turned to the Sleeping Beauty transposon, which had been shown by Ulrike Munderlo and Tim Curti to work in this system. So Matt designed a plasmid that has Sleeping Beauty inverted repeats for transposition and encodes a transposase, and then has the CAG promoter driving DS-RED, and that's flanked by ADP sites. So in contrast to the artificial exon approach that we'd used with Drosophila mosquitoes, we do expect that free-floating plasmid in this case is going to result in DS-RED expression. So initially on, we see a lot of cells that have the DS-RED in them. That's going to decrease over time as the free-floating plasmid gets lost. And what Matt was able to observe is small clusters of cells that remained DS-RED positive. That gave us the hint that indeed we were getting rare but real transposition events. Matt and his group then did a number of enrichments by fax over a course of 3 months in order to identify a pure population of this. And they were able to use a molecular approach to map the end of this insertion in a region in the Azotes genome. So we did have confirmation, we have molecular confirmation that it's in fact has inserted as in a transposition event. So again, that enrichment process happened, and we're very pleased to have what may not seem like the most exciting image, but was very exciting to us to have this enriched population. But of course, one of the open questions in a system like this where we feel like we can't take much for granted is, okay, we think we have ATP in there. Can we, in fact, actually use it for RMCE, suggesting that we will be able in the future to insert guides that way? So again, Matt and his team took on this challenge. So he developed a plasmid, in this case, using CAG driving GFP flanked with ATB. This free-floating plasmid is going to express GFP initially, but by the end of the assay period of day 36, you essentially have no GFP left because that free-floating plasmid, in the absence of an integrase, has simply disappeared from the cell population. By contrast, in the presence of an integrase, we get that initial population, but we have an enduring population that is GFP positive, consistent with successful RMCE. And so I find this particularly striking at day 36 here. We have less than 1% of cells scoring as positive by flow cytometry without the integrase, and that number is 10% when the integrase was present in the assay. So we feel that we have now demonstrated that we have a cell line that has ATP sites useful for this guide integration. And we next wanted to tackle the idea of adding a source of Cas9 and identifying promoters and making sure they're active. So Matt turned again to the Sleeping Beauty approach to add Cas9, used a similar approach of having, in this case, a BFP marker, waiting out the period in which the free-floating plasmid is lost, and we see a small population that are positive for BFP that we think are Sleeping Beauty transposition events, multiple rounds of FACS enrichment, and he now has a population of cells that are positive for DS-RED and for BFP, suggesting to us that they have both that original ATP cassette and should be expressing Cas9. In terms of the TIC-Pol3 promoters, Enzo Mameli applied the same pathway approach, or the same bioinformatics approach he had for mosquitoes. So he identified small nuclear RNAs in the tick genome, identified upstream of those candidate U6 promoters. The team cloned 4 of those promoters and have been subjecting them to a Cas9 activity assay. So that assay is described here. So we're starting with these cells that have ATP sites and have the Cas9. we're introducing a free-floating plasmid that notably does not have AdB. We're not trying to kick out the the DS-Red with recombination. Instead, we're interested to target DS-Red with the guide. So each of the candidate U6 promoters was put in one of these plasmid backbones so that a candidate U6 promoter will be driving a guide either targeting DS-Red or targeting using a non-targeting guide for this species. And then separately, we have a Pol2 promoter driving GFP so that we can identify cells that have received this plasmid. So Matt and team transfect this plasmid into cells. They then sort and ask among cells that are GFP positive and BFP positive, so they should have the candidate U6 guide and they should have Cas9, what % of those remain DS-Red positive? So again, we can compare non-targeting to targeting of DS-Red. With a non-targeting guide, we see what we retain this large population of DS-Red positive cells. And I think it's pretty striking here by contrast, when we're targeting DS-Red with the guide, with one of these candidate promoters, we see the absence of that population. Codify this in a number of ways. I'm showing you another way here where we're separating out the different data from each of these 4 candidate promoters and comparing them to what turns out to be more or less a negative control using the Drosophila Pol3 promoter. So in this case, we basically don't see reduction of the DS-Red positive population, but with each of these tick Pol3 promoters, we do see reduction. So this puts us excitingly and very recently on the path to feeling like we've tackled each of the major steps we need to tackle in order to establish large-scale screening in tick cells. So our next steps are to establish assays that will be appropriate in this species, to conduct some small-scale pilot screens, eventually then build a genome-wide library, and certainly our hope is that we'll be able to facilitate unbiased genome-wide screens in this health-relevant species directly. So I'm going to end here with a few final comments. So one thing I hope that I've convinced you of is that these CRISPR cell screens, the state-of-the-art technology that we're working on now in terms of screening platforms, are powerful and they're unbiased tools useful for gene function discovery. Another thing I hope I've convinced you of is that this is feasible to do in standard labs, and I'll say that we are currently working with a number of labs, both local and non-local to our region, and doing all we can through advice, protocols, materials, and so on to try to get them up and running with these pooled screening in their own labs to study topics of interest to them. And then finally, I hope I've convinced you that these CRISPR cell screens also open the doors to doing cell-based screens in non-model species in an exciting new direction that capitalizes on what we've learned technologically from Drosophila, but then applies it in a new way. So finally, I can't emphasize enough the extent to which what I've talked about has been a team effort, and I'm joined in all of this by an incredible team of people, both from the past and the current group, and we're also a relatively large group, as you see here in the DRC Paramount group. Norbert Paramount sets an incredible stage for technology development, encouraging technology development and optimization, and encouraging community outreach. I talked about some of the work that Claire Yanwei Hu has done in informatics. I would encourage all of you to check out what recent tools they have launched for the community. The URL here at the bottom right, if you put a /tools, you can reach an overview page of that. Jonathan Zirin handles all the in vivo stuff that I haven't had any time to talk about, and does an incredible job managing that side of things, directing that side of things. And then specifically the information I talked to you about, I showed you that Ram Viswanatha has really pioneered these screening technologies. Enzo Mameli was then key to extension to to mosquitoes, and Matt Bitnaro, together with Shrishti Goswami and Liam McKenna, have led our tick team in doing the work that I described in that species. I also talked specifically about work together with Hugo Bellin at Oz Kanka at Baylor, with Min Dong at Children's, and Joe Pedra at the University of Maryland Med School is our wonderful partner in the tick research. and Ryan Smith has been our partner in the large-scale genome-wide anomaly screening. We have a number of ongoing collaborations, and I just, will finally thank funding and some additional groups, including the DGRC, for their support of our efforts. So I'll stop there, and I'd be happy to take questions if there are any.
Well, thank you very much, Stephanie. That was amazing. Whirlwind tour of high throughput screening in Drosophila. While we wait for questions, yeah, Yuan Feng, can you go ahead?
Okay, sure. Hi, Stephanie.
Hi, Yuan. It's good to
Yeah.
see you. Another
Yeah.
former DRC person.
Yeah, thank you for the wonderful talk. So I just have one very basic question regarding the pooled CRISPR screen.
Mhm.
Probably I've heard like multiple times in the previous meeting, but I just completely forgot. So the thing is, how do you guarantee that one cell would just get one sgRNA there in the pooled screen? So the resistant phenotype that you observe is not from like synthetic phenotype due to multiple genes function.
Yeah, so that is a question we got a lot, and I did completely gloss over that. I think part of the answer is ultimately we don't, right? So we do think that there's more than one copy of this APP cherry in the cell line, and so it is possible to get more than just one. We don't think there's necessarily one per cell. What we can imagine, however, is that this is a numbers game. So we're transfecting lots of cells and looking at lots of data. The chance that the same pair gets into the same cell is small. So we expect that they're each going to get counted as positive, say, but like if there's one that's real and one that's not real, like in terms of association with a phenotype, the not real one is going to be different every time if you in terms of combination with a real hit. So I'm not sure I did a great job of explaining that, but I hope that makes some sense. Where again, like it's going to be at random what pairs are there. And so the one that truly has a phenotype is going to consistently be over or underrepresented, whereas anything that comes along is going to be random and it's not going to show up in that.
Okay, that makes sense.
Great.
Yeah, thank you.
Tamannash, do you want to go ahead?
Yeah, sure. This was a fantastic talk. I've really been following these resources for a long time. I have a couple of questions. The first one is, so for like human genome and like, people, when they do CRISPR screens, they order these guide libraries from like GenScript or something like that, right? So is there such like a similar kind of thing that's being headed towards for these model and non-model systems in insect cells?
So for good and bad reasons, I'm not aware of CRISPR library, like genome scale libraries for any of the species that I've talked about. And partly, I think that's because the community that are using these resources would be small, so it's not particularly attractive as compared to a company building a library for human use. That said, we are absolutely dedicated to helping others use these and gain access. So the version 1 of the library that we have is available from AdGene. We're partnering with people openly with the version 2 that we would recommend to use now. So you can simply send me an email and I can get you connected with the right folks to talk about doing that. And it's certainly our intention that once we've fully validated the non-model species libraries, we'll also make those available as well.
Thank you. Great. I have one more question, if that's okay. So regarding the previous question regarding the single ADP site or multiple ADP sites in the per cell, is it possible to like score those or sort those out based on cherry expression to kind of see like whether they're single or multiple integration events, or do they scale with expression of the copies present in each cell?
Yeah, I mean, the expression levels are kind of at least somewhat variable, even within a population that we had at one point single cell clones. So we think there's a single, set of events or event in there. So that makes me think that it wouldn't be quite as simple as we would want it to be in that regard. I think it's also possible that wouldn't necessarily be stable. So when what we've seen when we make CRISPR mutations, for example, if like we're making just a single gene knockout cell line, when we sequence that over time, we could see that basically one mutation seems to kind of take over. So we think there's probably a recombination event or some kind of, molecular biology event that's sort of biasing towards replication of one of those or repair with one of those or something like that. So I don't know that it would necessarily be stable over time or easy to detect. Certainly when we first get them, we're not going to take the brightest one or the dimmest one. We want something, in between, just because we expect that those cells will be a little bit more healthy. Yeah, I mean, it'd be really nice to know with complete confidence, but the flip side of that is we know it works, right? Like we know we jump in and do the genome-wide screens and are able to see interpretable data. So, it's a balance there. I also want to address about your first question. So I talked about the fact that we do at the DRC, we're doing RNAi in arrayed format and CRISPR in pooled format. There's nothing that would prevent doing CRISPR in arrayed format. And if you had a small number of targets, that might be possible. But speaking to your comment about commercial availability, we had conversations with commercial providers, and it would just be cost prohibitive to do. So we were really excited about the pooled screen format, in part because of cost. So I didn't put this on a slide, but it's actually considerably cheaper to do a pooled screen than it is to do the RNAi screens that we support, at nonprofit levels. So something like a tenfold difference if you're looking at genome-wide screening. So when I talk about these being accessible to individual labs, I mean that from a financial standpoint as well.
Thank you.
Okay, I'll go with one more raised hand before I go into the questions in the chat. So Julsana.
Yeah, can you hear me?
Yep.
Yeah.
Yeah. So the similar kind of techniques would be used for the yellow fever mosquito, right? Could benefit a lot of the.
So I'm sorry, I'm not hearing you well. I think I caught yellow fever mosquito. Could you repeat the question?
I mean, similar kind of techniques could be used with the yellow fever mosquito as well, like for isolating the cells of Aedes aegypti, and it could benefit like a larger population globally. That's what I feel, right?
Yes, complete agreement. And we are working to establish the full genome. So we did establish multiple species of mosquito. If there are cell lines available, there's no technological reason to expect that we couldn't establish it in others. We're also very happy to empower other labs to, to do the same things, to set up in new cell lines or in specific cell lines that they know have particular, virus amenable properties and such. We'd love to have more hands so that we can accomplish those goals. Yeah. The other thing I'll mention is, like, I had never looked at how many different species are covered, within insects by cell lines, and the number is like at least 200. So another area that we're looking at now is in crop pest insects. So many people are for example, familiar with SF9, that cells that often get used to express proteins, and they turn out to be, at least this is news to me, they turn out to be derived from an important crop pest species. And so that's another area in which we're looking. So Daniel, should we take this question from Lucy, or did you have something else in mind?
There are a few. We'll come back, Lucy.
I was just going to say,
are...
I think you just answered my question, Stephanie.
Okay.
Fantastic talk. It was about insect cell lines for crop health.
Great. Thank you.
Well, there are a few questions in the chat. I'll read them out. So the first one is from Sam Jang. Great talk, Stephanie. Could you expand a bit on how the screen can be adapted to aging study?
Oh, I may have put the wrong NIH grant there because it's not aging. I'm so used to NIGMS. This is terrible. It's allergy infectious disease. I should have been reading NIAID. So like that should now make sense that that's the institute that's helping us fund the tick project. So I apologize. It's in fact not an aging study. So hopefully you can come up with some answers to how this could be used as a format to look at some cellular machinery that are relevant to aging. I do not have an answer to that.
Okay. Scott has raised an hand. Scott, can you go ahead, please?
Hi, great talk. I'm wondering about how generalizable the results are even within Drosophila or between Drosophila cell lines, right? There's different genes expressed, they have different copy number, and different chromatin states. And I'm wondering if you have any plans to try in KC or more distantly related Drosophila cell lines to see how consistent the results are.
Yeah, so I love this question in part because it harkens back to Lucy Cherbas's work, where she has looked at those transcriptomes, and we have that data, as you may be aware. So certainly, yeah, any given cell line from Drosophila seems to express only about 50% of the genome. So the CRISPR activation is exciting, and that in a sense it gets us around that because you're looking for, it doesn't have to be expressed in the native state in order to identify it. But the basic answer is yes, there's every reason we could establish this in other species. I mean, in other Drosophila cell lines, that is a goal. One of the cell line group that we're looking at most are these new cell lines developed by Amanda Simcox that should more closely mimic specific cell lines. So they're glia-like cells and muscle-like cells. And one of the exciting things about that, in fact, we collaborated with her to do RNA-seq on those lines and characterize some of the transcript signatures there. An exciting part of that is like the muscle cells, you can treat them with ecdysone and get them to do muscle-like things. They'll become binucleated and pulsing. And so there again, a very exciting possibility for establishment of the system. And we'd love to have people come and say, this is what I want to do, or reach out and say, we want to do that in our own lab, and we'd empower that. And it is a longer term goal for our sort of next 5 years.
Okay, thank you very much.
Yeah, and in fact, with those cell lines in particular, and with KC example that you mentioned, I should also note that there are already KC cells that in the DGRC made that already have ADP. So we're a step ahead on our checklist. And some of the cell lines that Amanda Simcox reported also, by virtue of the genotype she started with in vivo, also have ADP. So we may be a step ahead with some of those.
Great, thank you.
Yeah, in doing that, answering Scott's question, you also answered one of Dan Tracy's questions. So we'll move on to the next question. This is from George. Wonderful presentation. You mentioned that fluorescent cell lines are in progress, such as metabolic sensors and pathway readouts. Can you share which pathways/cell states are nearing completion?
I'm such a technological junkie. I'm not sure I know the pathway names. I'm more focused on, okay, which fluors are we using and such. I know that our general approach is when we can, we'll borrow from others. So a lot of what we're doing is just comb the mammalian literature or comb even the existing in vivo fly literature. And then we can make a safe bet that those same sensors are going to work in this system. We do need something that's separable by FACS, so like if it's a real subtle change or if it's a fret sensor, it may not be ready for, it may not be appropriate for this kind of assay. But I think there's a number of things that we can and are trying. Metabolism is an area of interest in the Paramount Lab, so that is one of the areas in which we're just sort of looking around to see what's available and trying some of those out in Drosophila cells and other species cells.
Okay, so this is just a general observation that I have in terms of RNA high. How high is the demand at your center currently? Is it kind of on its way out, or you still have labs coming up to do RNAi screens?
For RNAi screens, the answer is sort of both. Are we phasing it out, and are people still requesting it? So we're in an interesting position. If we were a business which thank goodness we're not, we couldn't be doing these experiments if we're not, then we would have phased it out already. When we were in the 5 and 10 years ago, we were able to capitalize on economies of scale. So the general approach with the libraries that was to make a lot of double-strand RNA and then aliquot it and essentially sell an aliquot to a different lab, right? Like I hate thinking about it that way, but at some level, there was a business element that was like that. If we are not able to get a number of labs using the same library resources, then we're not recovering that full cost, then it's, it's difficult to replenish and such. And there's also, as you get someone who's really good at things and people move on to graduate school and such. And so there's been some churn with staff as well. We are still absolutely supporting RNAi screens. And long-term, what we will be able to continue to support are what we call cherry picks for people who are familiar with that. So we have a collection of the DNA PCR templates that were generated as template for in vitro transcription. So that covers 2 to 3 per gene for the whole genome. And those are very stable in a freezer. And we have liquid handling automation that we can pull them out, spin the plates, pick a subset of 2 or 3 or 200 and provide that to a lab that can then use it as templates for IVT. We're also continuing to support custom IVT. So if people want a 96-well plate of double-stranded RNAs, we can do that first step that I just described and then take it further doing the 96-well format IVT. That's in fact been one of the ways in which people are following up on CRISPR screens. So that gives us, like an added motivation for keeping that. And those have remained services that, it's not at the same pace, but we still I get requests for that, and we're still very able to predict costs, transfer costs, like do those in a reasonable way and continue to capitalize on the infrastructure here. That said, it's getting harder and harder for us to keep up with the maintenance steps with the double-stranded RNA libraries that we like, the genome-wide library and our focus subsets. In part because we have so many focus subsets, it's hard to know which one is the winner. We've had people interested in different ones. We'll have to replenish those again, like at large scale 96-well, in order to then replenish those libraries to sort of make aliquots that again get sent out. That's the harder service to maintain, and our current our next period of grant funding does have a plan for phasing that out. So I would welcome people to talk to me. We do have a lot of double-stranded RNA in the freezer. It's just sort of like, well, we're not sure whether we're going to be able to give you all the quadrants or not. So we're trying to make the most of what we have, but it is getting harder and harder. So it will be.
And segueing into cherry picks for requests for gRNAs, have you had anything like that coming through?
So we can't cherry pick from the pooled library.
I know. Yeah.
So the requests then tend to go to the TRIP team that does, of course, have to have separate library or separate guide that's going to go into separate fly. Jonathan occasionally gets requests for those and can fulfill them. We certainly, my awareness of it is it might be a couple a year. It's not been something that there's some huge demand for individual guides, in part because it's so easy to order a couple of primers, throw them into Blackbones that you guys make available and that other groups make available, and just wholly do that independently of us.
So the last comment I have is from Shafali, who says, great talk, Stephanie. Just wanted to say this is a beautiful talk, and which I completely concur with. Thank you very much. And so there's an additional question from Sam. Is there a tool to express sgRNA conditionally?
So I'm thinking of work by, not from our group, but from Philip Port's group at the DKFC in Germany. So I think that some of his approaches are amenable to Gal4 UAS-based control in vivo of guide expression. In terms of the cell system, I'm not aware. We're using U6, and that's going to be constitutive for the POL3. The way that in vivo Philip Port designed to get over that is to separate guides with tRNA-like sequences that then get cleaved. So you're sort of making in the cell, you're making those guides. We haven't tried that in cells. We haven't had a need for that. And it's very simple to make short oligos and clone them into these libraries.
Yeah,
I hope that answers the question. Yeah.
Yeah, Brian is chiming in saying that conditional Cas9 would be better, which does exist in terms of you can do tissue specific Cas9 if needed.
Yeah, certainly in vivo. We can do kind of leaky and strong using the metallothionein promoter in cells also.
So again, thank you, Stephanie, for a wonderful presentation, and thank you all for joining us and lovely interaction. If you can all turn your videos on, we'd like to take a picture of everybody here virtually.
I also want to extend my thanks to the audience for being here.
I got all the snapshots. So yes,
Okay,
excellent.
great.
Mhm.
And one last request, in case we're going to keep doing this going forward, and if you have any suggestions for speakers, please send it to dgrc@iu.edu. We're going to be running this biannually every year and hope to have similar interactions in the future. Yeah.
Bye, everyone.
Right, thank you again.
Thanks, Tom.